Now let's take a look at problem 7.16. So it has been suggested that any male determining genes contained on the Y chromosome in humans cannot be located in the limited region that synapses with the X chromosome during meiosis. What might be the outcome if such genes were located in this region? Okay, so I think we can take a just quick look at this. I don't think it's too complicated. So we have, this is problem 7.16, and we are looking at the Y chromosome. And it might be a good time now to just review the structure of the Y chromosome. So we have a small P arm and a longer Q arm. That doesn't really make much sense, right? The P arm is always smaller than the Q arm. So right here at the tip of the P arm, and the Q arm, we are going to have pseudo-autosomal regions. So PAR for pseudo, autosomal regions. And just quickly here, so this part down here, this is our heterochromatic region of the Y, and this is our euchromatic region, where we have somewhere between 50 and 75 genes. We don't know exactly how many there are in there. And then up here we have our very important region called the sex determining region of the Y. So SRY. And within the SRY region is the SRY gene, which makes the SRY protein. Okay, in the SRY gene and protein, this is the key gene which determines maleness on the Y chromosome. And a lot of the others are important for fertility and sperm development. So fertility, sperm development, and I don't really know of any other functions for Y chromosome genes. There may be some in there, but I don't know. I don't know that. I don't think anyone knows that just quite yet. All of the region between the pseudo-autosomal regions is referred to as the MSY, male-specific region of the Y. Okay, now the question is asking us, for all these male-specific genes, these 50 to 75, how come they're not in the pseudo-autosomal region? And the reason is, these are the, so if we have a meiosis here, right, all our chromosomes have to line up on the metaphase plate. Let's say this is chromosome 1, and we have a little chiasma forming between those. This is chromosome 1. Chromosome 2, 3, 4, all the way through 22 pairs of chromosomes. They all have to line up there, and that leaves either just two X's in a female meiosis, but an X and a Y in the male, and we need to have these aligned so they can segregate to different secondary spermatocytes during meiosis. So here's our X chromosome here, and to be honest, I don't exactly know what parts of the Y chromosome actually pair up. No, I know what parts of the Y pair up with the X chromosome. So they are going to be these regions down here, the pseudo-autosomal regions. And I'm assuming that they are matching the same ends of the X chromosome, but that might not be true. So these right here, sorry I drew this or diagrammed it so small. So these are going to pair. So we can get crossing over between the X chromosome and the pseudo-autosomal regions of the Y, wherever they're homologous. That's where we can get crossing over. And this is these are the regions that have to pair, which allow these chromosomes to pair during meiosis so they can align together on the metaphase plate, and then they can segregate away from one another during meiosis. Now, we can get crossing over between the pseudo-autosomal regions, and that is why we can't have a male determining gene within the pseudo-autosomal region, or a male-specific gene, like SRY. So if SRY was actually in the PAR region, well then SRY can jump over to the X chromosome during crossing over during meiosis. And in some cases that appears to happen when we have unequal crossing over, it's called, and we don't need to worry too much about that. But when unequal crossing over occurs between the X and the Y, sometimes the SRY can move over to the X, and that can lead to the XX male syndrome. Okay, so this is why we don't want male determining genes or male-specific genes in the pseudo-autosomal region, because they can cross over and end up on the X chromosome, and they're not useful on the X chromosome because they're for males, and sometimes they can cause something like XX male syndrome, especially if it's an important gene like the SRY.